Good afternoon, children. Last time in our class, we had started with our topic in lesson number 2, that was the female reproductive system, and we had used many new terms and this very important diagram of the anatropous ovule. The notes I have already sent in a file, and I hope you all have written and understood and tried your best. Today, children, we are going to start with the formation of this oval structure, what we call by many terms. Embryo sac is the most frequently used term, but we also call it as megaspore or megagametophyte or a female gametophyte. Now, this structure, which is our new topic for today, is to be understood by one of these possible pathways. The first and foremost, suppose this embryo sac is to form. Now, what happens? You have to just look up to this cell. This is a very primitive or an early part of structure which will be developing as an embryo sac. You're going to study this. This is just a diagrammatic layout through another textbook I am showing you for understanding. No need to draw it. This is the Newton textbook. Now what happens? One of the cells in the subepidermal or the hypoepidermal region, it functions as an archisporial cell. See, this is a mass of parenchymatous cells, as you just now you have seen this over here. But one of the cells, it becomes functional as the archisporial cell due to two reasons. Why? Because this has dense cytoplasm and a prominent nucleus. This is one of the subepidermal cells of the nucellus. Remember nucellus? This is the nutritive tissue. Now, it undergoes one of these pathways. Let us go one by one. The first pathway, which is more common, is that the archisporial cell directly functions as the megaspore mother cell, right? This is how the cell directly functions as the megaspore mother cell. Now what happens? This cell undergoes meiosis or the reduction division and ultimately forms a linear arrangement of 4 haploid megaspore cells. You can see very clearly these are a linear arrangement of 4 haploid. All you can see is half the number of the mother cell. So this is 4 haploid megaspore cells that are formed from the megaspore mother cell. Now what happens is that look at this position of the nucleus, children. This is towards the micropylar end. The nucleus is towards the micropylar end, and ultimately when this arrangement of 4 haploid cells are formed, 3 of the micropylar cells, they degenerate, right? Mostly the top 3, that is from the micropylar end, they degenerate or wear away. But the last one, what is this region ultimately? What is opposite to micropyle? Yes, it is chalaza. So the functional cell at the chalazal end now becomes functional, right? While the other 3 have degenerated. So this functional cell with the haploid nucleus now undergoes mitosis, right? With 2 nuclei, as you can see, they have moved to the opposite poles, forming a 2-nucleate embryo sac, right? Now further, 2 more mitotic divisions have to occur. First was already done. Remember children, here it was meiosis. First it underwent meiosis, giving rise to 4 linear arrangement of haploid megaspore cells. But now 3 had degenerated from the micropylar end, and the lower part of the functional cell, now it becomes the active one and underwent mitosis. So what is the result? 2-nucleate embryo sac was formed. Second mitosis, 2 becomes 4, that is doubled up. And now finally a third mitosis, giving rise to 8 cells. Count it, 1, 2, 3, 4, 5, 6, 7 and 8. So ultimately children, it is the 8-nucleate embryo sac that is formed. Alright, so ultimately with a slight rearrangement of cells, this is how the structure is formed. Count it again children, 3 at the top, 2 at the center and 3 at the bottom. So 3 plus 2, 5 plus 3, 8. So this is ultimately how the female gametophyte is formed in the first method from the archisporial cell. So what is this formation of the female gametophyte called children? It is called as megasporogenesis. So what is the definition of megasporogenesis? The development of megaspore or embryo sac or megagametophyte or female gametophyte from the megaspore mother cell, what we call as MMC. I hope you understood. Once again, before we go to the second method, I told you one of the epidermal cells, it differentiates from the rest of the nucellus as the archisporial cell because it has dense cytoplasm and a very prominent nucleus, undergoes directly as a change into megaspore mother cell, and this is the micropylar end. So the nucleus is basically towards the micropyle, divides by meiosis, giving first to 2, this is the dyad that is formed, and then ultimately to 4 megaspore cells. These were haploid. The first 3 towards the micropylar end degenerated, and the lower one that is towards the chalazal end, right? This becomes functional, undergoing further to mitosis. Through mitosis giving rise to 2 nucleate, ultimately 4 and 8 nucleate embryo sac, right? The other pathway, which is less common, this is when the archisporial cell, right, undergoes periclinal division. Remember children, I had told you the meaning of periclinal, which means parallel to the surface. The division is parallel to the surface. Ultimately, as a result of this division, 2 cells are formed. We call outer as parietal cell and the inner as porogenous cell. The parietal cell further divides into parietal tissue, whereas the inner sporogenous cell now behaves as megaspore mother cell. That is, this megaspore mother cell here, it worked directly, but here it is now through an alternative step of this method. And the other part would be the same. Ultimately, again, it undergoes meiosis, giving rise to structure like the previous. So what is the difference of previous method with this second method? Yes, it is the only step which is this. This step is extra because here the archisporial cell directly functioned as MMC. Here the archisporial cell, before it becomes as MMC, is undergoing a periclinal division. This step is the only difference. The rest, product or the outcome of both the divisions are the same. 4 cells they were formed here and 4 cells they were formed here. The rest of the further journey would be same. That is 3 degenerate, 1 remains functional, and that undergoes mitosis, then a second mitosis and a third mitosis, giving rise to 8 nucleate embryo sac. So I hope children you have understood this journey of division, what we call as megasporogenesis or formation of embryo sac or this structure. Now we have 3 types of embryo sac. We call them monosporic, bisporic and tetrasporic. Monosporic is the most common type of embryo sac. First, let us understand the meaning. Mono means 1, bi means 2, tetra means 4. Now what happens, like let us refer to this diagram. You have seen that out of 4 cells, 3 degenerated and only 1 was functional. So when one functional cell is differentiating or becoming the embryo sac, this type of embryo sac, where the mother cell is 1 cell, will be called as monosporic, right? But sometimes what happens, the embryo sac is formed from this dyad. Dyad means a group of 2. So when these 2 cells, they turn into an embryo sac, this will form a bisporic embryo sac. And very rarely what happens, all 4 cells, they are formed after meiosis. All 4 they form the embryo sac, and then we call it as tetrasporic. Once again, monosporic, as I told you, is the type of embryo sac which is formed from 1 cell. Where is this 1 cell coming from? The tetrad, right? The megaspore tetrad, 3 worn off, that is degenerated, and only 1 was functional, which changed into embryo sac. So this is monosporic. The second I told you bisporic. Bi means 2. So when this dyad, this changed into an embryo sac, it is called as bisporic. Similarly, when this tetrad changes into an embryo sac, that is all 4 cells are involved, then it is called as the tetrasporic embryo sac. So that is very easy. Moving further, now with the structure of the female gametophyte. Just now you have seen children the development of female gametophyte. Now let us see the structure. The structure means you're going to talk about this arrangement of 8 cells, right? I'll just show you in your textbook. This is what you're going to study. This is the mature embryo sac. You can see those 8 cells. 3 are at the chalazal end. They're called as antipodal cells. 2 are in the center, what we call as polar nuclei, which later on fuse to form a secondary nucleus. And 3 are at the bottom, right? The central one called as the egg cell or the oosphere, whereas the 2 side ones, they're called as synergids. Now there are again many new things to learn, children. The embryo sac, you can see they are made up of 3 parts. Antipodals, number 1. 2, the polar nuclei. And 3, the egg apparatus. Egg apparatus means this assembly of 3 cells. Now what happens? This egg cell, let us begin with the egg cell. Let us come to our diagram. Here, this egg cell is polar. Why it is polar? Because you can see the nucleus is towards the chalazal end. Chalazal means the end opposite to micropyle, right? So if this is the micropyle, the pollen tube are going to come from over here, then the opposite side would be chalaza. So you can see clearly the nucleus is at the chalazal end, that is at the top end. Whereas at the micropylar end, you can see vacuolated cytoplasm. There are many vacuoles that you can see. So this becomes polar body, right? And then surrounded by the 2 side cells, what we call as synergids, right? These synergids, they have a structure like this in each of them. You can see a finger-like structure, that is filiform apparatus, which further divides into what you call as microfibrils or small, even smaller finger-like structures. You can see each, one over into this and the other over here. So what do you call this? Filiform apparatus, which ends up into microfibrils. Now microfibrils, they're very rich in polysaccharides. Remember polysaccharides, the form of carbohydrates or a source of energy, right? Now the filiform apparatus along with the finger-like structures, they're called microfibrils, right? With the finger-like structure, they're called microfibrils, and they have a very important function, that is they guide the pollen tube to the synergids. They're not just for no reason. If they're here means they're a source of carbohydrate, and they're going to guide the pollen tube, which is going to come and take the destination complete over here. All right. Then coming to the only diploid structure in the center. One polar nucleus that was here. If you come back over this side, see. 2 polar nuclei are there. One has been shifted from the top, the other has been shifted from the bottom, and both of them, they have fused. Now after fusion, you're going to call them as secondary nucleus, the only diploid structure. And what is the fate of it? It will end up forming the primary endosperm cell. So 2 things I have completed. Just now I told you about the egg cell or the oosphere, which was bipolar, that is nucleus at the top, and then the vacuolated cytoplasm towards the micropylar end, synergids along with filiform apparatus and microfibrils I have told you. Now coming to antipodals. Antipodal cells at the top, which are assembly of 3, they are very rich in enzymes, starch, lipids and proteins. They take an active part in absorbing nourishment from the nucellus, and at the end they degenerate. So children, each structure has a function to play, has a role to play, right? Antipodals also, they're not just like that. They're here to absorb the nourishment from the nucellus, that is the nutritive tissue. Then secondary nucleus will form into the endosperm. Then filiform apparatus, I told you, they have the guiding part for the pollen tube to enter, that is through the synergids, and this is what they are. Let us see the other functions of synergids. Synergids, they absorb nutrients from nucellus, secrete active substances for attracting pollen grains, act as a shock absorber for the growing pollen tube, and they form the seat for pollen tube discharge. So synergids, they have a lot of role to play, right? And herein the egg cell or the endosperm is ultimately the most important part, which will be later on discussed during fertilization process. That is, this is one of the places where the male gamete will come through the pollen tube and will fuse here. right, to form the zygote. So every part here, as I discussed, is important, right? Before we move further, children, everything on this page I have discussed with you. Only one thing remains here, polygonum embryo sac. What is a polygonum embryo sac? This is the type of embryo sac we call as polygonum. Why? Because look at the cells. Top 3, they were haploid, N, right? Bottom 3, they are all haploid, again N. But only the second one, third one, that is polar nuclei in the center, this is the diploid. So into one assembly only, embryo sac, you can see haploid and diploid both are present together. Such an embryo sac we call as polygonum. I have written over here and I'll be sending you through the notes. So this part, children, I have completed with the development of embryo sac. I hope you understood through 2 journey. Most common was this one and lesser common is this. Then I told you the structure of embryo sac and the importance of every structure, their functions. I have written and put an arrow clearly and written under them only. So please make your notes accordingly. This is just the gist of your textbook also in a very systematic manner. Now moving further to the types of ovules. This is again a new topic. Before we see the types of ovules, children, I'll show you there are 6 types of ovules that are present. This one we have understood last time only, the anatropous. The anatropous is this B1. We have discussed last time if you remember, but we'll again discuss it. So what are the names of the 6 ovules? Let us look at this diagram and understand. Orthotropous, first one, anatropous, hemianatropous, where you can see that this has become a transverse body, right? Then campylotropous, which means this has become like a horseshoe shape, right? Turned like a horseshoe shape. And then the 5th one, where it is called as the amphitropous, and the last one, which is called circinotropous, right? So let us see the characteristic of each of the ovule. Now, first one, I'll be reading through the notes. Let us see through the diagram. The first thing is that orthotropous is found in walnut and polygonum. This is the most primitive type of ovule where the funicle is straight, ovule lies straight, and funicle, chalaza and micropyle, they are in one line. Let us look at this diagram. What is funicle? Funicle, let's revise because most of you must have forgotten in our last class. 3 terms again I told you, important for you to revise before we move further. Funicle was this short stem, right? What was hilum? This point of attachment. And what was raphe? The attachment of funicle with the integuments, right? So now let us see. Here it is saying the first type that the funicle is straight. Look at the funicle. Yes, it is straight. Then ovule lies straight in the ovary and funicle, chalaza and micropyle are in one line. So you can see the funicle, the micropyle at the top and the chalaza, the portion opposite, they're all in one straight line. This is very primitive form. Second is the anatropous ovule. This ovule is the most common type seen in gram and pea, and the body of ovule is inverted. You can see that the funicle is over here, small structure, and this whole thing is inverted. The micropyle lies close to the hilum and the funicle is curved at the apex forming raphe, right? Now what happens? Finally, the micropyle and chalaza are in one line. Unlike the previous, here you saw funicle, chalaza and micropyle all in one line, but here it is not. Only micropyle and chalaza in one line. So this was the anatropous. Coming to the third, the hemianatropous. This is seen in families like Ranunculaceae, Brassicaceae and Malpighiaceae. Ovule lies transverse, or is like laid down horizontally at right angle over funicle. You can see if this is funicle, children, then the ovule is forming a 90 degree with the funicle, right? And this structure, it is over the stalk of funicle, which is attached to ovule somewhere in the midway. The micropyle and the chalaza lie in a transverse line. Unlike the previous, they were in a vertical line. You can see them. Micropyle and chalaza were in a vertical line, but here they are again in straight but transverse line instead of vertical. Next, the fourth type, the Campylotropous. This is very clearly seen in Capparis and mustard. What is the special feature of this type of ovule? It is curved. You can see the ovule is curved or bent like a horseshoe shape, right? In such a way that the micropyle, chalaza and funicle, that is micropyle, chalaza and funicle, come to lie at one pole, and funicle does not fuse with the integument. You can see that the funicle is nowhere fusing with the integument. Coming to fifth type, Amphitropous. Amphitropous seen in examples like Capsula and Lemma. The body of ovule as well as embryo sac are curved. You can see that. Ovule is also curved and the embryo sac also is curved. Right? Now, ultimately what happens is that the hilum, that is this point of attachment, the chalaza over here and the micropyle, they are all in close proximity. Once again, before we come to the last one, into this Amphitropous, the body of ovule along with embryo sac is curved. Then hilum, that is this point of attachment of funicle with the body, the micropyle and the chalaza, they are in close proximity, but they're not in one line. Finally, the Circinotropous, seen in family Cactaceae, that is Opuntia or what you call as cactus. Ovule is straight. You can see this. Ovule is straight with micropyle facing upwards. Micropyle, which is most of the time here down. You can see this here, here and here. Right? Micropyle was down. Here it was transverse, but only here it was up. So here micropyle faces upwards, right? And funicle is elongated and coiled. Look at this funicle. It is much elongated and coiled, completing and encircling the ovule. That is why it is called as Circinotropous ovule. Right? So this entire thing, children, in a summarized way or whatever I have explained, I have written into this table. Please pen it down as a summary to complete the type of ovule. The most important and very important with diagram is the Anatropous ovule. Moving further to our new topic of pollination. Pollination, we know, children, already from our previous learning, it is the transfer of pollen grains from the anther to the stigma, and it is very commonly seen and only seen rather in gymnosperms and angiosperms, right? Then this means the plants with the naked seeds and this means the plants with covered seeds. Pollination is broadly of two types, self and cross. Self-pollination is further of two types, the autogamy and the geitonogamy, whereas cross-pollination we can understand by the term like xenogamy and allogamy. Let us see the differences. Before we move further with the terms, let us see the differences in a pictorial arrangement first. Look at this diagram. This is a plant, right? And it is showing bisexual flowers. In one method, you can see that the pollination is going between the same flower, like a male is fertilizing the female of the same flower on the same plant. This would be called as autogamy. I'll show you more clear diagram into our own text. Look at this over here. This is autogamy. That means the male fertilizes a female on the same flower on the same plant, right? Geitonogamy means when the male fertilizes the female between two different flowers but on the same plant, it becomes geitonogamy. That is, whether you say auto or geitonogamy, both mean self-pollination because the parent plant is same. But when you say cross-pollination or allogamy, means the pollination between two flowers on two different plants. That is, male fertilizes the female which is on another plant but of the same species. Right? So that is called as the xenogamy or allogamy. Something like this. This was auto, this is geitonogamy, and this is the xenogamy or allogamy. Further to study, autogamy is achieved or brought by two methods, homogamy and cleistogamy and chasmogamy. Homogamy means when the aerial flowers are there and open flowers are there. For example, potato, wheat, rice, mirabilis, and catharanthus, etc. Right? So into this, the only method is homogamy, means aerial and open flowers. But the cleisto and chasmogamy, very important terms, children. What does it mean? What does they mean individually? Cleistogamy means underground flowers which remain closed throughout their life. Let me show you a picture of a plant here. This is a picture of a chasmocleistogamous plant of Commelina. You can see there are two types of flowers. At the shoot region or the region above the ground, there are normal chasmogamous flower, whereas underground flowers called as cleistogamous. So what are cleistogamous flowers? Imagine, children, flowers which are born under the soil, and these remain closed throughout the life. They never open. Very small, inconspicuous. That means they can hardly be noticed. And obviously they are not scented, bright, colorful, but they are rather very colorless, odorless, no scent, no nectar, nothing. That is, they are least attractive, nothing to attract the insect for pollination. So what is the only method of pollination in such flowers? Yes, it is self-pollination. That is, there is no agency to invite the insect. Coming to the aerial flowers, these are the chasmogamous flowers. Now what is special? They are aerial. Unlike this cleistogamous which were underground, the chasmogamous are born in the air. They are like normal flowers. They are large, brightly colored and nicely scented, right? They can fertilize or pollinate by both methods, that is self-pollinate or cross-pollinate. But later on, they become cleistogamous. That means these are different from homogamy, or they are going to become later on like a underground closed flower. So have you understood the difference, children? Once again, the cleistogamous flowers, just now I have shown you, they are underground flowers which remain closed throughout life. They are small, inconspicuous, colorless, odorless, and only self-pollination, right? Whereas chasmogamy is a method where aerial flowers which open like normal, the large, brightly scented, then brightly colored, and both self and cross-pollination, whereas these ultimately become like these. That is, the aerial become like underground later on. And why does this happen, children? This is to ensure fruit formation, right? Which is in the interest of the plant. Then there is a term we call as chasmocleistogamous flower. Chasmo and cleisto means plants which bear both the types of flowers. Just now I have shown you. This plant, Commelina, the complete term is Commelina bengalensis. This plant has both the types of flowers born on the plant, and therefore it is called as the chasmocleistogamous plant. So these are the flowers which are having both the types. Examples: Commelina bengalensis, Viola, Oxalis, Arachis, etc. And next time, children, I'll be starting with another topic. Though you have done this in class 9th, but we'll be starting again with a fresh learning, adaptations or contrivances to ensure cross-pollination. This I'll be starting with you tomorrow. So today, what you have your assignment, children, please start writing down from here, draw them accordingly, try to label them very in a very nice manner, and go through the terminologies by heart. You should not have any confusion. And 2 diagrams you must practice very nicely. One is this anatropous ovule, which I told you is extremely important, and the other is these types of ovules you should identify by their diagram. If it is vertical, or if it is transverse, if it is inverted or coiled around with the covering, etc., then what are their names? And another important, very, very important diagram for you to draw as a homework and learn is this embryo sac. So please draw and label it very nicely. Thank you for just now, and keep revising nicely.